and probably get started 5 minutes early. And our first session is organ aging and growth signaling. And our first speaker is Ana Valencia, who is a postdoctoral fellow in the laboratory of David Marcinek. Ana, take it away.
Hi, good morning, everyone. I'm going to share my screen here. Okay. Thank you, the organizers, for putting this together. Shelby, Alessandro, Mitchell, great work. And thank you for the invitation. So yeah, like Alessandro said, I'm a postdoctoral fellow in Dave Marcinek's lab. And like many of you here, we're interested in maintain healthy aging. I'm sorry, I'm just trying to, oh, there we go. That's one of our main goals, like many of us here. And specifically, my lab is interested in the interaction between mitochondria and cell stress and its effects on the pathology of chronic disease and aging. And specifically, I am interested in understanding the impact of nutritional stress in the context of aging physiology. So with aging, we normally see a decrease in resilience. So with aging, we see an increased susceptibility to stressors. We clearly see this now with COVID-19. And like I said, I'm particularly interested in this nutritional stressors that can include diets that are, for example, high fat, high sugar diets. And this bar graph is just to show you the number of papers that have been published on aging and these diets. So there's a lot that has been done on high fat and very little on high sugar. So today I have two stories for you, and I'm going to, using these diets, and I'm going to start out with the one that is not commonly known. So let's start with sugar. So the first question for me was, is the response to a high sugar diet different between young and old mice? And specifically, I come from a muscle physiology background, and I was curious to see if the sugar diet could accelerate sarcopenia. So this diet, we gave young and old mice high sugar diets, specifically 60% of the calories coming from sucrose. So that's a lot of sugar. And you can see on the right, on the red bars, you have the animals consuming a high sugar diet. So we have the young and the old. And you can see there's a slightly increased energy intake from this diet. So now we get to the interesting parts. So body weight changed during this week. The young mice tend to increase in weight, while the old mice, there's a little bit more of a variability in its effects on body weight. In fact, we wanted to do this intervention a little bit longer. So we did have some mice consuming this for 4 weeks, but eventually they did not eat and they lost a significant amount of weight. So according to our iCook, we had to cut that group out. But just something to note, they did lose weight after 4 weeks. So the most interesting finding here was not in skeletal muscle. It was actually in the heart, which was unexpected. Hence, I'm talking about it. So we found that the old mice consuming a high sugar diet had bigger hearts than their chow counterparts. And in fact, even those mice that we had to euthanize, those 4-week-old, their hearts also tended to be bigger, even though they lost so much weight. So that's something to note here. So when we think about sugar and heart, this is not the first study to show that sugar induces cardiac hypertrophy. This has been shown in young mice throughout a longer intervention. So this specific study did a 20-week intervention with fructose in the water. And you can see here on this rectangle, the hearts of the fructose-fed mice were bigger. And also on the right, we see the cardiomyocytes were bigger. And using a mitochondrial targeted antioxidant, SKQ1, it was able to protect from the fructose-induced hypertrophy. Another study did a similar thing where it gave fructose through the water. And here on the right, we see that the heart is bigger. And they also show that mitochondria actually become swollen. And so here, these two studies show that mitochondria may be a mediator in the cardiac hypertrophy induced by high sugar. So then I wanted to conduct an experiment, focus on the heart, and I wanted to see if I could replicate my previous findings. And I also wanted to treat the mice with SS31, which it's a drug that you will hear later on today as well. So very briefly, this is it's a synthetic peptide that co-localizes to the cardiolipin in the inner mitochondrial membrane. And it appears to improve the organization of the complexes, and it reduces redox stress, specifically in mitochondria from older animals. So here in this study, I had young and old mice that were given saline as control, or SS31, through an osmotic pump for 2 weeks. And during the second week, they were given this high sugar diet, or chow. On the left, we have, on the top panel, we have the hearts of the young mice that are pretty much the same size despite of the group. And on the bottom, we have the hearts of old mice that are bigger. And especially the high sucrose saline group, the hearts were bigger. So if we look on the right, again, I was able to replicate my findings where old mice consuming a high sugar had bigger hearts, but not the mice treated with SS31. And on the bottom, we did do echocardiography to measure a left ventricular wall thickness. And this is before and after. So here, the only ones that changed significantly were the mice consuming, again, a high sucrose diet. A big question when it comes to changes like that in the heart, we want to know whether there are changes in cardiac function. So like I said, we did do some echocardiography. Jeremy Whitson did this from the Rabinovich lab. Now he is an instructor in Davidson's College in North Carolina. So sorry, but he did great work here. So on the left, we have the eta-A ratio, which is an indicator of diastolic function. We see a reduction in diastolic function in aging mice, but no effect with the diet or SS31. And here on the right, we have indicators of systolic function, ejection fraction and fractional shortening, where we saw no differences. So despite these changes in heart size, we did not detect differences in cardiac function. Because mitochondria appears to be a mediator in this effect, and SS31 did have an effect, we wonder if cardiac mitochondria could play a role and if ROS production was affected. So we isolated mitochondria from these hearts and measured ROS production using Amplex Red. And while there are differences between young and old, we did not detect differences between the diet groups. We also wanted to see, we wanted to characterize this hypertrophy more. So we wanted to see if it was a difference in cardiomyocyte size that we could detect. And using H&E stain slides, we measured the size of the cardiomyocytes, but surprisingly, we did not detect differences in any of the groups, not even between young and old, despite the differences in tissue weight. But one of the advantages we can do with these slides is to count the number of cells to give, and this is not a direct marker of inflammation, but it could indicate a level of inflammation. So we did this systematically, counting the number of nuclei in per stained area. And we did this in, systematically in image J. And interestingly, we found that both the young and the old mice consuming a high sugar diet had more nuclei, which could be indicative of inflammation. And something interesting is that here we see an effect happening in the young. So now work in progress to actually see if this is inflammation that's happening. We're doing some gene expression. So here are some hypertrophy markers on the left and inflammation markers on the right. And this is still underpowered. This is a work in progress. We need to add more animals to this. There's 3 animals per group right now. So I can't draw any conclusions from this. However, I just wanted to show you so far what we have. There tends to be an increase in hypertrophy markers in the old, as well as inflammation, but we haven't detected differences yet with high sugar. So to be determined. So in summary, if you were still, trying to get your coffee and everything, let me just tell you, 1 week diet increases heart size, but it does not worsen cardiac function. And cardiomyocyte size does not explain the difference in organ mass. And 1 week of high sucrose increase the number of cells in the heart, which could be indicative of inflammation. And we're trying to figure that out. Unexpectedly, we found that the old heart is very sensitive to high sugar consumption, and that it only takes a week to see a difference. So we found that high sugar is actually a really bad stressor for aging mice. And the questions are, is sugar promoting inflammation? And we have a lot of other tissues. We have liver, we have adipose tissue, we have pancreas, kidney. So if anyone is interested in collaborating or anything like that, please feel free to send me a message. Okay, so my next story real quick is high fat in aging. So this is the other diet that has been commonly known to induce stress. And this particular diet is 60% of calories coming from fat. Just to show you here, this diet, we actually gave them exercise for 4 and 12 weeks, so a much longer time. And here on the top, we have food intake in grams on the left and kilocalories. So here we see that there's an increase in calorie consumption in the beginning, and then it levels off to the same level as the chow. And on the bottom, we have the changes in body weight. As you can see, there's a dramatic rise in body weight in the first 4 weeks, but then it levels off a little bit after that. And then the chow at the bottom, you see a decreasing body weight over time, which is very normal with these aging mice. And again, I should say that these mice are 24 months old when they start the diet. Same with the high sucrose study. So in terms of body weight, so this is looking at mice, the 4-week and 12-week treated mice. Not surprisingly, we see that the mice fed a high-fat diet are heavier. Their hearts, especially at 12 weeks, they're bigger. Their fat depots, visceral and subcutaneous, are also larger. And livers tend to be also heavier. So this is not surprising. But again, my original plan was to see whether this diet could accelerate sarcopenia, so back to skeletal muscle. One of the methods that we use that is great to look at changes in force over time on the same mouse is using this setup by Aurora Systems that allows us to measure force of plantar flexion. So this is kind of when you raise your heels, so plantar flexion, and by stimulating the tibial nerve, this takes away the whole voluntary part of muscle contraction, so we can control and actually achieve, get the mouse to contract maximally. So maximal force, we can measure on the same mouse over time. And here we have the longitudinal changes in force. In the chow, you see in the black that there's a decrease in force over time, showing that the mice are becoming sarcopenic. But something very surprising is that the mice fed a high-fat diet did not have a reduction in force. When we look at this cross-sectionally, there is some variation between the mice. Some are stronger and weaker. So at 12 weeks, we don't see statistical difference, but it tends to go in that direction. Another thing that we can do with muscle testing is that we can test muscle fatigue. And this is by stimulating the muscle every other second for about 2 minutes. And you see in the green that the green is a little bit higher up, meaning that they're more fatigue resistant. So when we actually measure the fatigue index, so a greater fatigue index being more fatigue resistant, we see that high fat really promotes fatigue resistance in this mice. So overall, the whole thing about fat being bad for muscle contractile function, that didn't really fit in. It protected the mice from sarcopenia, and it also made them more fatigue resistant. So then we also wanted to look at the size of the gastroc. Here on the left, we see that the gastrocs were bigger in the high fat fed groups. And specific force was not changed. So the changes in force are because of change in mass. And here, because I only have a little bit time here, there were increases in respiration, mitochondrial respiration in these mice. And a big thing is I want to see if it would induce redox stress. And surprisingly, no. We looked at glutathione and protein carbonase and plate assays on H&E, and there were no differences between groups. So unexpected, the high-fat diet does not increase redox stress or decrease mitochondrial respiration in old skeletal muscle, and high-fat diet appears to benefit skeletal muscle contractile function. So one of the questions of future directions for this is want to know how high-fat diet is protecting this muscle, maybe through the neuromuscular junction, or either promoting hypertrophy or preventing atrophy. I really want to thank my lab and Jeremy and Rudy for helping me with this project, and all of you, and thank you for listening.
All right, fantastic. Thank you, Anna. Very interesting talk. We already have a couple of questions. There was one that arrived through the Slack channel. Darian Bug is asking, hi Anna, your talk is really interesting so far, although you don't see changes in myocyte cross-sectional area in your H&E stain. Have you looked into isolating CM, I think it's cardiomyocytes, and doing, I'm not sure what this is, L by W ratios in length by width ratio, I guess, and area in your high sugar studies? I would be curious to see if it is remodeling the cardiomyocyte aspect ratio rather than just cross-sectional area. I have found this to be the case in some of my heart failure models. Also, have you looked into the fibrotic deposition in your hearts? Yeah, length to width. Yeah.
Okay, thank you so much. Yes, I would love to talk to you. Yes, regarding, no, I have not done that. All I have done is looking at these slides and looking at cross-sectional area. And we particularly measure cross-sectional area where there was a nuclei in the middle, because I know that there can be variation along the fiber. But it is something I would be interested to talk to you about. In terms of fibrosis, I have done a Picoserius red staining on these slides and did not detect a difference between groups.
Okay, then we had a question from Matt, actually. And Matt, do you want to unmute yourself and repeat it, or you want me to read it?
Sure, I can do that, I guess. So, I guess the question, well, the observation is that there a lot of people in the field are focusing on protein as the bad guy when it comes to aging in the diet, and I'm wondering if you think that protein restriction might suppress the effects that you see from a high sugar diet on cardiac hypertrophy, or from a high fat diet in muscle hypertrophy, and then as a follow on, you know, might rapamycin do the same thing?
Yeah, absolutely. So that's an interesting question. So the diets that I used did have the same amount of protein on there. But you're saying maybe if we reduce the amount of protein in addition to adding sugar, if it could be protective.
Right, exactly.
Yeah, I think so. I think some of the things I would have to do to be more certain of that is looking at, you know, the mTOR pathway to see if those things that are activated that normally, you know, extra protein also activates are the same, to see if reducing protein could maybe improve that. And the same thing for rapamycin. But I think so, it's possible.
I mean, part of the reason why I ask is I feel like a lot of the studies on protein level in diet in the context of aging, and particularly as people try to extend that to human relevance, you know, most of the data on protein consumption in people are in people who are overeating, right? And so I wonder about this balance between protein in the diet and the consequences of a high caloric diet, whether that's sugar or fat. The other question I had is, did you look at any sex-specific effects? Because we know that sometimes, you know, with these aging interventions, at least, there are very strong differences in males and females.
Absolutely. And no, I have not looked at females just yet. I guess I wanted to have a more set of things that I wanted to look at with the males and then see if it happens also in the females. But I expect, especially with the young and old individuals, to see even more of a difference in the females, especially because females, when they're young, you know, they have more estrogen, tends to have more antioxidant capacity. And when with aging, that is gone. So it would be something interesting to look at.
All right. Then we have also on the slide is Kara Green. Hey, Kara. Hi, Anna. Awesome talk. I'm sorry if I missed it, but for the first part of your talk, were the control and high sucrose diet isocaloric? If they were you reducing another macronutrient to make it isocaloric? I was wondering if the high sugar diet induces hyperphagia. And actually, she thinks that Matt's question has answered most of her question, but if there's anything you think you want to add to her specific question.
So they are not, so we just had a chow diet that we normally give to our mice, and the high sugar diet was experimental, and they were very similar. I do have the information somewhere, but they were similar in composition, not exactly very isocaloric. I think the difference was very small, but generally, yeah, that was the difference. The young mice did increase in body weight, but the old mice did not. So it is not, while it has more energy, it's not like a high fat diet that has a lot of energy. So they're more similar in that sense.
And I'll sneak in my quick question here. At what age do you think we should stop eating dessert?
What age? Well, I mean, apparently that's hard to say, right? I think when it comes, something very important is that when you're looking at diets, you're looking at an everyday regimented consumption of this nutrient. That is different to what we do as humans. So it will be interesting to see if having perhaps sugar a different, every other day or something would have the same effect. So I'm not going to give up on cake yet.
All right. So one of the advantages that we start a little bit early is that we still have a couple of minutes and we have another 3 questions. Mitchell, I think you had a question if you want to ask it real quick.
Sure, absolutely. Hey, Anna, yeah, that was an awesome talk. So I don't know a lot about cardiomyocytes, but I think they're one of the cell types that undergoes multinucleation and polyploidization. Mm-hmm. So I was curious if you looked at that as an alternative hypothesis for the immune cells coming in, or that inflammatory response.
Mm-hmm. No, no, but it's something definitely that I have to keep in mind. Yes.
Yeah. I was trying to think of like the best way to do it, like maybe like a flow-based assay or something. Mm-hmm. That would be the best way to catch it.
Yes. And then that to the first question, it would look more at the area of the fiber as well. And you can see the number of nuclei. Absolutely.
Yeah. And then relating that to other cell types that go through multinucleation, do the liver cells, like the hepatocytes, do they also show the same thing? Is that something that sugar's doing, is kind of spurring on the need for more DNA?
Yeah. Thank you, Mitchell. That's a great idea.
All right. Then we had anonymous attendee. I have checked off the anonymous questions, so all questions from now on will have your name. But yeah, this anonymous attendee is asking whether there was a change in the blood pressure of the old animals fed the high sugar diet.
I wonder the same thing. Unfortunately, we do not measure blood pressure, but it is something that is possible because clearly this is a stressor. The fact that the heart is getting larger, maybe because, yes, there's increased blood pressure or some other system that is, there's something going wrong and the heart just feels like it needs to get stronger or something like that. So it is possible. It's hard to measure do those measurements in mice, but it's something that's still done, and it'll be very interesting to find out.
All right, and we're gonna do one last question, and then I'm gonna redirect all other questions. I saw that somebody has posted a question in the chat, so please type it in the Slack channel or in the Q&A tab. But the last question for you to answer live is from Jeremy Whitson, who's asking whether you have considered formation of advanced glycation end products as the mechanism for the heart's sensitivity to high sugar.
Yeah.
I would think that the level of age would almost certainly increase and harm cardiac function.
Yeah, thank you. Hi, Jeremy, and thank you for your hard work on this. Actually, yes, I have done some Western blots looking at end glycation aged or AGEs, and I did not find a difference. I know there's plate assays also that you can do, but I did not see a difference with high sugar, which I was surprised with.
All right, very well. And thank you so much, Anna. Fantastic talk. I'm gonna move on.